In the next section, we'll begin our discussion of neurophysiology and the generation of the action potential. In this portion, we'll talk about neurophysiology. We're going to focus on the interactions between neurons, and these are the important elements in the brain which allow communication to occur between certain parts of the brain and other parts of the brain, and between the brain and the rest of the body. From a therapeutic perspective, it's important to understand how neurons communicate, but because without this understanding, we would not be able to offer particular treatments which are directed towards altering the activity of particular parts of the brain. One example of this kind of a therapeutic approach is deep brain stimulation. In deep brain stimulation, particular areas of the nervous system are targeted for alteration of their activities. It is only through the understanding of the normal activities of particular parts of the brain can we so specifically be able to go in and manipulate the activities that are abnormal. In the case of Parkinson's disease, we have deep brain electrodes which are placed in what's called the subthalamic nucleus, and the activity of subthalamic nuclei neurons are altered by the activity in the deep brain stimulators. Here is an example of a patient who has Parkinson's disease who has a deep brain stimulator implanted, but the stimulator is turned off. If you observe with the video running that this subject has a very typical Parkinsonian walk. He has a slowed, stooped, and shuffling gait. And one way of quantifying the severity of his gait disturbance is by counting how many steps it takes for this subject to turn around. In contrast, when the stimulator is now turned on, one can observe this subject now walking down the hall with a greatly improved walking speed and a greatly reduced number of steps to turn around. Again, the important point to recognize here is that these types of therapies can only be made available through the detailed understanding of how neurons communicate with each other. And that's going to be the focus of the current part of the lecture, and that is how do neurons communicate. Shown in the next slide is a generic neuron. So neurons are, as I mentioned before, the communicating elements in the nervous system. And neurons are like other cells. They have a cytoplasm, they have a nucleus, they have a membrane. But they also have specializations that allow long-distance communication to occur. On the receiving end, they have branched cytoplasmic extensions known as dendrites. And we'll talk about these in more detail as the lecture goes on. And on the opposite end of this cell, what's shown here is a long axon. This is a long cytoplasmic extension that allows these cells to send signals to either other neurons or other organs in various parts of the body. Axons have some specializations that allow this to occur. They have myelin in many cases and what are known as nodes of Ranvier and synaptic swellings, and we'll talk about the details of these structures later on in this lecture. Neurons can be placed into one of several structural classifications based on the number of projections that come off of the soma or the cell body. Cells that have more than 2 projections coming off of the soma are referred to as being multipolar. This actually corresponds to the majority of cells in the nervous system, the majority of neurons in the nervous system. Unipolar cells have a single projection coming off of the cell body, and bipolar cells have 2 projections coming off of the cell body. These cells are found in restricted portions of the nervous system. Unipolar cells are found in the dorsal root ganglion, and bipolar cells are found in certain sensory epithelia, such as cells in the retina, cells in the 8th cranial nerve, as well as cells in the olfactory bulb. Within the subclass of multipolar cells, there are multiple other subclasses. Alpha motor neurons in the spinal cord, shown on the top left, are a form of multipolar neuron. as are Purkinje cells, which are found in the cerebellum and shown here on the bottom left. These cells have a very unique structure such that they have a tremendous amount of branching occurring in the dendritic tree, but all this branching occurs in a single plane. Multipolar cells are also found in the olfactory bulb. These are a different kind of cell than the bipolar cell previously described. These mitral cells have multiple projections coming off of the cell body, as well as a tuft coming off of a single dendrite. These are known as mitral cells. And on the right, we see the majestic pyramidal cell. Pyramidal cells are found in the neocortex as well as the hippocampus. These have a cell body which is shaped like a pyramid, hence the name. There is typically a single dendrite coming off of the base of the pyramid and multiple dendrites, both apical dendrites coming off of the top of the pyramid and basal dendrites coming off of the other portions of the pyramidal cell body. Neural transmission itself utilizes something akin to a Morse code, and the actual blips in the Morse code are known as action potentials. These blips are ultimately small fluctuations in the voltage across the membrane, and these fluctuations are dependent upon a new kind of channel that we've not yet spoken about, known as the voltage-gated sodium and the voltage-gated potassium channels. Shown on this slide are the people who discovered the mechanisms by which these action potentials occurred. That's Hodgkin and Huxley, and most of their early work was done in the squid giant axon. The portion of the neuron where the action potentials are generated is known as the axon hillock. This is the spike initiation zone, and the reason that action potentials form here with the lowest threshold is because this part of the axon has a very high concentration of voltage-gated sodium and potassium channels. The action potential itself refers to this fluctuation in the voltage across the membrane. So shown here is a voltage versus time plot. The blue line represents the voltage as it changes over time. And at rest, this particular cell has a resting membrane potential of about negative 90 millivolts. Shown on the bottom left is one of the voltage-gated sodium channels described earlier. These voltage-gated channels have two gates. They have an M gate and an H gate. And because they are voltage-gated channels, these gates will open and close as the voltage changes. When a cell with these channels is depolarized, this depolarization will cause the M gates to open. As the M gates open, sodium will rush into the cell through these gates. As sodium rushes into the cell, it will depolarize the cell further, which will cause even more M gates to open. This triggers a positive feedback cycle, because depolarization opens up M gates, and M gates will trigger more depolarization. As more and more of this occurs, there will be an explosive depolarization of the cell. And that transition point is illustrated by the dotted line known as the threshold. When there's a transition to this rapid influx of sodium, the cell reaches a point where the permeability for sodium actually exceeds temporarily the permeability for potassium, and the voltage moves very near to the equilibrium potential for sodium. As a matter of fact, the voltage will surpass zero. This is known as the overshoot, and will stop near to the equilibrium potential for sodium. At that point, the second gate, the H gate, will close. The H gate in the sodium channel is also voltage gated, but its kinetics are slower than the M gate's kinetics. Therefore, it will close the channel, but it will close the channel after the M gate has allowed in enough sodium to allow this depolarization to occur. A second kind of voltage-gated channel is the voltage-gated potassium channel. And the voltage-gated potassium channel has a single gate known as the N gate. And the N gate and the M gate behave very similarly. That is, they will open up when cells depolarize. The major difference is that the N gate has much slower kinetics than the M gate. Therefore, the N gate for potassium channels will open up far later and does not begin to open until after the overshoot of the action potential. When the N gate opens, however, it will allow potassium to leave the cell. And so many N gates are open and so much potassium leaves that the cell will repolarize and it will actually undershoot. That is, it will cause the cell's membrane potential to approach the equilibrium potential for potassium and become more negative than the baseline or resting potential for the cell. This fluctuation, the depolarization followed by the repolarization, is known as the action potential. And cells can fire thousands and thousands of these action potentials over time. This will ultimately deplete the sodium and potassium gradients as sodium rushes in and potassium rushes out. And these gradients are repleted using the sodium-potassium ATPase shown on the right. Now, if one wanted to trigger another action potential during the course of this action potential, the response of this cell will vary depending on specifically when that second stimulus is given. If a second stimulus is given in this yellow box, this period during the action potential, another action potential will not fire. This is known as the absolute refractory period. The reason the cell will not fire is because the sodium channels, the voltage-gated sodium channels, are not in the appropriate configuration to allow another action potential to be triggered. That is, the sodium channels are not in the M gate closed, H gate open configuration. It is not until these channels regain their basal configuration of M gate closed, H gate open that another action potential can fire. If, however, a stimulus is given after that point, another action potential may fire because the sodium channels are back into their native configuration. However, if the voltage is more negative than the baseline voltage, it will require a larger stimulus in order to reach threshold. Therefore, this period is known as the relative refractory period. That is, it takes a relatively larger stimulus to achieve threshold than it took before the action potential occurred. This next slide reviews the channel gating. So shown at the top is the tracing of an action potential, and shown at the bottom is the change in permeability for sodium and potassium throughout the course of the action potential. And also shown are cartoons of the sodium and potassium channels and their behavior during the course of an action potential. At rest, the sodium channels have their M gate closed and H gate open. And at the initiation of the action potential, the sodium channels have the M gate open to allow sodium to enter the cell. At the peak of the action potential, the H gate closes. This is known as the inactivation gate, and no more sodium will enter the cell. And ultimately, after the action potential, the sodium channel will regain its native configuration of M gate closed and H gate open. The potassium channel will begin with the N gate closed. As the cell depolarizes, the N gate will open and will remain open for a protracted period of time to allow a large amount of potassium to leave the cell, causing the cell to hyperpolarize temporarily, and ultimately the cell will achieve its baseline potential once the cell's voltage-gated channels return to their native configurations. Shown at the bottom is the change in permeability of potassium and sodium. G and A refers to the conductance or the permeability for sodium, and G sub K refers to the conductance or the permeability for potassium. The dotted line refers to sodium, the solid line refers to potassium. As shown for sodium, the sodium permeability at baseline is lower than the permeability for potassium, As the action potential ensues, the sodium permeability temporarily exceeds the potassium permeability and then returns to its baseline permeability as their channels return to their native configuration. Potassium, by contrast, begins with a permeability which is higher than that for sodium. Later in the course of the action potential, the end gates open, the permeability for potassium rises, and then slowly drops back down to baseline. One important concept to understand about action potential generation is the all or none hypothesis, and that's illustrated here. On the left, you observe the response of a small step stimulus in a neuron. This stimulus is too small to elicit an action potential, and the voltage tracing for this cell is shown in the pink box on the left. With a slightly larger stimulus shown with the blue box in the top middle, this stimulus is now supra-threshold. That is, it's strong enough to allow this neuron to pass threshold and to fire action potentials. If a larger stimulus is given, one does not get larger action potentials. Instead, one gets more action potentials, and that's shown on the right of the pink box. So the important concept to know here is that larger stimuli are encoded by the nervous system as more frequent but not larger action potentials. So then, to review the major properties of the action potential. An action potential is a depolarizing event which involves progressive depolarizations that ultimately reach threshold and trigger the action potential itself. The action potential is a reversal of the membrane potential followed by hyperpolarization and then a return to the resting membrane potential. And importantly, you either get a full action potential or not. Once you reach threshold, you get the full action potential, and stronger stimuli only result in more frequent action potentials. And then finally, it's important to recognize that action potentials only occur in so-called excitable cells. That is, cells that have voltage-gated sodium channels, which only comprises 3 cell types in the body: neurons, cardiac muscle, and skeletal muscle. This concludes our introduction to neurophysiology and the generation of the action potential. This concludes our introduction to neurophysiology and the generation of the action potential.